Blood and urine are the most common fluids that we're going to get in the lab, but we have to be aware that all these other things come in. It's really important for you to know what they are when they come in. So if somebody brings in something called pericardial fluid, you know what you're looking at, because it's going to make a difference with how things work. So different other body fluids include things like amniotic fluid, spinal fluid, tissue, sputum, serous fluid, sweat, semen, gastric secretions, nasopharyngeal secretions, and saliva. These are all other types of fluids we're going to see in the lab. So we're talking about cerebrospinal fluid, and these are all links. These are in your PowerPoints. You can take the links to go to different videos that'll like show you what's kind of like some video of like what's going on, types of collections and stuff. But what happens with spinal fluid, it's called cerebral spinal fluid because it literally flows through the brain and up and down the spine. So the fluid we find here is the same fluid up there. It just is constantly flowing throughout. So when we have something that's going on in the brain, instead of actually doing a brain tap, we can actually do a lower spinal fluid tap and see what's going on in that whole system. It's all one continuous system. So what they do is they do a lumbar puncture, and they put a small little needle into between the lower vertebrae, very similar to a spinal tap or a nerve block or an epidural, where they put the needle in, but instead of putting something in, they're going to take some fluids out.
It does not feel small.
It does not feel small, no. But they use numbing stuff, and it's definitely gotten better over the years than it used to be. Because
my nerve.
Hmm?
They hit my nerve with the FV.
Yeah, they got mine with my last MACE thing too. My leg kept flipping out. They're like, "Yep, we need to move that," because it was the crazy. I'm like, I'm trying to hold it, and the nerves weren't going to let it. So things do happen, but they've gotten a lot better with what they do. So they put this small needle in, and it is a very small needle, and what happens is the fluid drips out the back. So they actually collect the fluid as it's dripping out, and they get 4 tubes. They're marked 1, 2, 3, and 4. They're supposed to collect them in order, and then the tubes get sent to different departments. So the first one is likely to have some red cells just from the traumatic tap, right? It went through skin. So we don't want to do a cell count on that one because it's going to have a falsely elevated amount of cells. So that one usually is going to go to either, actually that one goes to micro because we've cleaned the skin, and the first one should be the cleanest. I think it goes to micro.
Is
Is the second one on micro?
It goes to chemistry.
First one goes to chemistry. Okay.
Traumatic tap.
Traumatic tap. Okay, so the first one goes to chemistry. Second one goes to micro then, after we flushed it.
Yes.
Third one gets cells.
Yes.
And then the fourth one, I think, is just to hold.
Yes.
For other things. So by the third one, we've gotten that traumatic tap cells out. And so you get these tubes, and each of them only have like a cc in them. We don't want more than that. If we pull too much fluid out of that area, you can end up getting really bad cranial headaches because of the fluid coming out. So it's really important they don't take too much. And then when they close it off, it's really important that they hold that pressure and stop the bleeding at this point because we don't want the spinal fluid seeping out afterwards. The other reason they go so low is you'll see the spinal cord, it goes down to here. So like the spinal cord at this point, this is the spinal cord. They don't want to hit the cord. They want to go below the cord when they're doing this. Okay, so that's cerebral spinal fluid. Pleural fluid, pericardial fluid, and peritoneal fluid. So over here, it's just kind of a general what you're thinking about when you're looking at these. It's a serous cavity. There is a reason that the organ, whatever the organ is in a serous cavity. If it was not, for example, the heart, if the heart did not have a fluid sac around it as it pumped, I'd end up with a lot of friction. It would be constantly rubbing up against all the other stuff that's in my chest, and you'd constantly be dealing with bruising and friction and problems. And how long do you think a heart's going to last that doesn't have that fluid around it, right? So it has that fluid, so as it's pumping and moving in a bag of fluid, which protects it. So for all these organs, we have a parietal membrane, a serous cavity with the organ in the middle of it. So when we are looking at pericardial fluid, pericardial, right? Go back to those root roots. Around the heart has a sac around it. So as it's pumping, it's in that fluid. If it gets jostled, if you get knocked to the ground, it's not hitting something hard. It's bouncing around in fluid, right? So that's pericardial fluid. Then we have pleural fluid. Pleural fluid is what wraps around the lungs. So it's the fluid around the lungs here. And then we have peritoneal. So all of these inside inside organs, your stomach, your intestines, your liver, all this stuff is what's in the peritoneal cavity. So it's all in one big cavity like that. So it's all protected. So when you're getting fluids from these areas, you need to know where it's from, because different ones are going to have different normals and different normal cells. So it's important for you to be aware. Pleural fluid, this is an example of a thoracentesis. So what happens is if you get too much fluid buildup in that pleural cavity around the lung, it's going to push that lung up, and you're not going to be able to get a full breath. Because a full breath allows the lungs to only open up and close up. But if something's going on, that fluid's pushing it, you're not going to be able to get that full breath and expand your lungs the whole way. So it's really important as that fluid starts building up that you get in here and you drain it. So this is an example of what's going on. It's a needle through the rib, back part of the ribs here, and it's going to drain that fluid off, which is going to allow that lung to fully expand back out. So that's how you remove pleural fluid. Usually it's in a bag. This is an older picture. We actually used to collect it in bottles, and we get the bottles in the lab. Thoracentesis is actually the name of tapping the chest cavity to remove the fluid. So thoracentesis gives us pleural fluid. Synovial fluid, this is an arthrocentesis, which is the removal of the excess fluid in a synovial joint. So synovial is a joint. So same idea, you get these membranes of protection, and sometimes if you get too swollen, something's going on, you gotta get synovial fluid. Somebody that might have gout, for example, or something going on where you see other crystals. What's happening in that fluid? We're gonna do an arthrocentesis to get that synovial fluid. Bronchial alveolar lavage. So what happens in this situation? A lot of times we get these because somebody has an infection and we can't figure it out. The sputum is not growing anything. And a sputum usually is either expectorated or a suction, where they go in and they pull that sputum out, trying to get it clean. Because when it comes through the mouth as it expectorated, what's our mouth full of? Bacteria, ick, stuff, right? So we're not going to get a clean sample. So they've gotten more where they do more suctions, but now if the suction's not clean, they can't figure out what's going on, they're going to do this bronchial alveolar lavage. Lavage means wash. So they literally put a bronchoscope down the throat, into the terminal bronchial, in the alveoli, they take this saline and they flush it, and then they suck it back up. So what they're trying to do is figure out what is going on in these alveoli that are causing the problems. When you get these in the lab, then they're going to test for mycobacteria, they're going to test for fungi, they're going to test for all sorts of bacteria, they're going to test for some of the bacteria that don't grow on normal plates. They're going to be doing all these different tests because now they got this sample of something really, really deep, and they're trying to figure out what's going on. This is somebody where they have pneumonia that they can't clear, and they can't figure out why. So they're going to do this bronchial alveolar lavage. I always joke they come in at 3:30 on a Friday afternoon, and it's a massive process. But when I was at the UW many, many years ago as a student, 3:30 every Friday afternoon, they're trying to get out the door at 4, and this 4-hour process would show up in the lab. Because you take the specimen and they require all this different processing. You do all these different slides because you're staining for all these different things because you're trying to figure out for this person what is going on. If they go to the point where they do a BAL on somebody, they've exhausted all other options. I mean, this is not something they do lightly. So, but that's what a BAL, when it comes in the lab, that's what you're looking at.
What part of the lab are you in to do those, to process those?
Depends on where you're at. Usually it's gonna be micro for these, because most of it's fungi and, they get sent out. A lot of these get separated out into different things and sent out to different places because most labs don't do it all at their facility.
So would that be done for something like Valley Forge fever, or I don't remember which valley it was.
Desert Valley, or the one down in Arizona.
Yeah.
Yes. This is how they would diagnose that a lot of times because they can't get it to grow otherwise.
Okay.
Yeah, that's why. There's always weird. I
I'm
don't
glad
want to call them
you
weird,
talking about
but there's
it.
those like really rare, crazy diseases that happen in this part of the country, or this part of the country, or over here, this country there. And so a lot of these diseases that are so rare aren't going to grow on a normal sputum. And if they can't diagnose it otherwise, this is probably how they're going to end up doing it.
Cool.
Amniocentesis. We will talk about amnio a couple times in blood bank next semester, But we use an ultrasound, we take this fine little needle to get some amniotic fluid to see what's going on in the fetus. A lot of times we're looking for bilirubin to see what's going on if we're dealing with a baby that's dealing with a hemolytic reaction, hemolytic disease of the newborn reaction. Mom's producing antibodies against the baby cells, so we can do an amniotic collection at that point. Semen analysis, I am going to show a video on this also. When you do a semen analysis, we're going to look for fertility usually. So there's two different ways of looking at this. We look at it when you're dealing with fertility problems in somebody, like a lot of families when they're trying to start their family, they can't get pregnant. So what we'll do is we'll do tests on both mom and dad and see what's going on. And we'll do fertility testing and semen testing to make sure. We want to make sure that there's enough of it, the viscosity is okay, and it's moving well. So we're looking at number of sperm, sperm morphology, and sperm motility. Because a lot of times what might happen is the morphology might have double-headed sperm. I don't have a picture, I think that's coming up in the video. Some of the sperm have double heads. Well, if there's two heads, they're not going to swim very well, right? Because they're going two different directions. Sometimes the tails aren't moving well, sometimes they're real sluggish. So it's important for us to look at motility. What is it doing? And then number, are there any there, and are there enough of them there? What's going on? The other end of when we look at sperm and do sperm counts is post-vac. So post-vasectomies, we have to make sure, did it work? Lots of people skip their follow-up appointment and end up pregnant a year later. It is very important if you or somebody you know gets a vasectomy that you do the follow-up. Okay, I've seen it more than once. I've actually seen, honestly, 10 years later, it grew back together. I have a friend. They thought they were done. 10 years later, she came along. So that's the other thing. So we see it for fertility treatment issues, like ahead of the game, like where's the problem? And then we see it afterwards to make sure we took care of the problem. Okay, so we see it on both ends. We would always get this in the microlab at UW, and we would do the volume and then send it out. Sperm do not live a long time outside of the body, so it's really important that you keep them at body temperature and get them to the lab quickly. So it's not something you can collect the day before and take in the next day, put in the fridge, because that's not going to tell us much. Other specimens I'm not going to get into: bone marrow, breath samples, throat swabs, hair, stool, tissues, lots of different stuff out there they can test for. Certain things you'll cover in different classes, like little bits here, little bits there, but we don't spend a lot of time on these specialized samples. If you land someplace that does specialized samples, you'll learn a lot more about them then.